Our next speakers hail from... Why universities always have these very long titles, but I'm going for it. The Environmental Microbiology and Molecular Analysis Laboratory in the School of Natural Sciences, Macquarie University. This laboratory explores microbial diversity in nature. They have used molecular techniques to investigate a range of organisms, from marsupials and insects to native and agricultural plants, down to fungi and bacteria in soil and water. Join me in welcoming Professor Michael Gillings and Vanessa McPherson, who are generously donating their time to unveil the intriguing life of fungi and their intricate connections with trees and the surrounding environment. Thank you.
Hi everyone, thanks so much for coming. It's a pleasure to be here, and so tonight Michael and I are going to be sharing with you some of the amazing things that fungi do in our ecosystems and why they're so important in terms of keeping our trees happy. So fungi are an amazing group of organisms. There are three main kingdoms that we're all familiar with of life on the planet. Those are animals, plants, and fungi. So these fungi here are kind of like the reproductive structures of fungi. So they're sort of like the flowers on a plant. So who in the audience has heard that fungi and plants are kind of similar? No? What about animals and fungi? So we know that animals rely very much on plants in the world, right? So fungi are very much reliant on plants too, because they're actually more closely related to us than they are to plants. Bit of a plot twist, huh? So fungi are actually really poorly known, and now that I've shown you some pretty pictures, I'm going to throw some statistics at you. So in terms of what we have named in terms of organisms on the planet, we are pretty good at identifying the large things. So chordates, anything with a backbone, we pretty much have most of them covered. So the things that have been named kind of closely aligns with what we think exists. In terms of plants, there are about 430,000 species, and we think we've named most of them. When it comes to invertebrates, they're kind of hard to catch because they're small and quick. So, there might be about 8 million species that are predicted to exist, and we think we've named about 25%. But if you look at the orange here, for fungi, we aren't even sure what we don't know. Like the estimated number of species that exists is anywhere from between 3 million to 8 million. Like, that's a huge range. We have no idea. We've named about 150,000 species, which would make up about 2 to 8% of that total. And most of those species that have been named are actually in the Northern Hemisphere. So Australia, just as unique as our plants and animals are to our biota, our fungi are probably just as unique. So why is it that they're so underdescribed, even though they're so beautiful? Well, they're really, really small and hard to see. So that's my fingernail in those pictures. And they are 3 different examples of fungi that you might find in the local area. And they're really, really small, but so beautiful. They're ephemeral, so they don't last very long. So that middle panel there, that's the same specimen at 6 a.m, 9 a.m, and 12 p.m. And then at 12:01, it's just a pile of black goop in the lawn. So they don't last very long, so they're hard to catch. And down here, this very last panel, believe it or not, those 3 pictures are all examples of the same species. So they're really difficult to tell just by looking at the morphology. And Michael will tell you a bit later how we know that they're the same species in the first place. And one of the most important things, especially at a tree symposium, to mention is that most of these fungi spend their life underground as mycelial networks. And as I mentioned earlier, these things that we're seeing in the photos, they're the reproductive structures. So most of what's going on, we don't get to see. So why is it that fungi are so important? Well, they're so important because they actually play a lot of different roles. They wear a lot of different hats. They're extremely diverse, and they play roles in breaking things down. So they're the saprotrophs. They hang out on the surface and they turn things over and make them accessible again for the next lot of organisms to use. They can be pathogens and infect plants or animals or other fungi. But perhaps most importantly, they are mycorrhizal. And this means that they form really important networks and associations with trees. And this enables them to exchange nutrients. So they supply plants with things like phosphorus and water and nitrogen and even sugars. But they receive sugars from those plants in return. So it's a real partnership going on. So the pictures here that you see of the fungi, they're examples of things that associate with eucalypts in our local area. So yeah, they're just amazing. And 85% of plants, so that's the majority, are dependent on fungi. There are some specialists, so things that can access their own nitrogen because they've got, like weird little tricks. So there are carnivorous plants like Drosera and sundews that you might have seen in the local area too. There are also plant parasites like mistletoes, but arguably they are relying on fungi because they're parasitizing another tree, which is relying on the fungi. And there are also things that specialize in like the extreme environments, so really salty areas or water or tree branches and things like that. And here is the thing we've all been waiting for, the Wood Wide Web. So this is a really famous hypothesis that talks about the interconnection of mycelial networks and trees. And if you've ever been walking through the bush and you find yourself in a dense canopy and there's not a lot of light getting down to the ground, but you might look around and notice that there are saplings that are managing to make a living. How is that possible if they're not getting access to the light? And it's because of these mycorrhizal networks that they're able to do that. So what's happening? As you can see in this diagram here, and this is a northern hemisphere example, but the large dots are the adult trees and the smaller dots are the saplings. And all of those lines are mycorrhizal connections between individual trees. So the reason that this is important is because the tall trees that have access to the light are able to subsidize their saplings through the mycorrhizal networks. So all of the photosynthate, so the sugars that they make from sunlight, are able to transfer to the saplings through the mycorrhizal networks. And in exchange, the mycorrhizae are making things like water, nitrogen, phosphorus, all those nutrients in the soil available to all of the plants in that ecosystem. So the importance of this is that fungi are central to sustaining generations of plants. It means that there can be ongoing survival of those ecosystems. And it also reduces competition between individuals and between species, so we can maintain a diverse variety of plants. So this is kind of a snapshot of the kinds of friendships that tree roots can have with fungi. So up the top left is what we call an ectomycorrhizal relationship. So the fungi are on the outside, ecto, of the roots, rhizal. So they're really closely intertwined and they're exchanging nutrients between each other. It's great. And down the bottom, we've got endomycorrhizal fungi. So these are really tight. So the fungi are actually inside the roots and they are exchanging nutrients really, really closely. And this is the level of closeness that our fungi and our trees have with each other. They're exchanging things not just at the cellular level, but on the inside the cell level. And now let's talk about regeneration, because that's a really important part of our goals in our local areas as well, and in being sustainable and making sure that we're looking after our resources, because they're amazing. So this is a study from Western Australia. Ectomycorrhizal fungi, which are the good guys. This red line shows you the baseline of what things were like before this site was used for mining. Those black dots show you the levels of the good fungi that were measured for 25 years after they tried to rehabilitate the site. On this side are the saprotrophs, so they're kind of like the bad fungi, so they're all the things that hang out on top and that are breaking things down, but they're not necessarily forming associations with trees. That red line is the baseline, and all those black dots are what happened after regeneration. So you can see this shift between the ectomycorrhizal, or the partnering fungi with the trees, towards things that decay, even after regeneration efforts. So it's possible that with bush regeneration, we need to actually also be looking at regenerating soil diversity and fungal diversity in order to make sure that the trees that we're putting into the ground have a chance at being resilient for future generations as well, and not just surviving for that one generation. So one of the examples of this is fungi. So orchids are very dependent on fungi. Often the relationship is so close that one species of orchid will be dependent on one species of fungus. And if they don't find each other, they can't survive. So Thelomytra atrax is one that we were really excited to find, and it's called Thelomytra atrax because atrax is the genus name of the funnel web spider, and you can see that the hood there kind of looks like the fangs of a funnel web. It has one leaf, as most orchids do. Calladenia transitoria in the middle there. That one is hardly open ever. We've seen it open a few times, but we could have missed that 5 minutes. That one on the end, it doesn't have a leaf at all. So you can kind of tell, even just by the amount of foliage that these plants are putting up, that they're kind of doing a little bit of work, but for the most part, they're actually parasitizing the fungus and making the fungus do all the work for them. But they're incredibly diverse and beautiful, and, you know, the fungi are keeping up with them. So, it's really exciting to see this coevolution of fungi and orchids, and there's just so much more to be explored and discovered. So some orchids are actually entirely parasitic, and they do none of their own work at all. So they don't have a leaf, they don't have chlorophyll. You can see here that these have no green whatsoever. This one here, the bootlace orchid, you can see that on an Angophora. And they're so unusual that we kind of don't even know what those associations are. Perhaps one of the rarest things we've ever found was the Eastern Australian underground orchid. So Rhizanthella slateri is so rare that it didn't even make the top 10 rarest plants in the world list. Its slightly more common cousin in Western Australia did. So we were slightly upset for this little guy. But it's just amazing. So it spends its entire life underground. It flowers under the leaf litter. When it does flower, it smells awful, but coffin flies think that's delicious, and they will go down and pollinate these flowers. And then when those flowers are pollinated and become fruit, they start to smell like vanilla. and bandicoots really like that. So they will go and eat those fruit and deposit them in a nice little packet of fertilizer somewhere. And if those seeds are able to find their fungal partners, they will germinate. However, the fungal partner requires a tree. We don't know what that species of tree is yet. But you can see here that if any one of those links is pulled away, this can't exist. So why did we start looking at fungi? Well, it was because of COVID, and we were locked out of the labs and we couldn't help ourselves, so we went into the bush and we started surveying all of the wild things out there. So we started in the Lane Cove Valley because that was, as far as we were able to explore. But we loved being out there, and we were just blown away by the beauty of fungi. They were just amazing, and we couldn't believe when we started looking into it how rare so many of these things are and how under-described they are. And a lot of the things that we do have here, they're so beautiful and breathtaking, and they belong here, they're ours, and we need to look after them and the things that they depend on. So here are just some examples of the things that we've seen in the local area. And all of these photos that you're seeing in this talk, they were taken on our mobile phones. So science is accessible to everyone. And all of these things have a role to play in our ecosystems. So these are the mushrooms and toadstools. These are sort of the more the traditional format of a mushroom, or what people think of when we say fungus. These are the waxcap fungi. They are beautiful. They range all of the colours. A lot of them are either unnamed or are only found in certain places. Like, for example, here we have the Lane Cove waxcap, which was discovered and named in Lane Cove, which is pretty exciting, and we've only seen it a couple of times, but it's been exciting every single time. So the non-gilled fungi, if it smells, if it is spiky, goopy, weepy, leathery, it fits into this category. Bolatellus emodensis over here, this beautiful pink. We often see it at the base of blackbutts or peppermints, so there's obviously some kind of relationship going on there. And Aceroe rubra, the starfish fungus, take this fact with you and share it around because it'll make you popular. But this was actually the first fungus to be described in Australia. So rather than the other way around, it was introduced to Europe rather than being introduced here. Also, it smells really bad, just a heads up. These are the ascomycetes, so these things are really, really small and you have to be looking really, really hard to find them. But when you do find them, they're just absolutely beautiful. This one here is one of my favourites, the bird's nest fungus. So those little egg-like things in the cup, they are actually the spores, and when raindrops fall into the bird's nest, they cause the little eggs to kind of splash out, and that's how they disperse. So these are the bracket and crust fungi. These are actually saprotrophs, so these are the things that are performing like the breaking down kind of tasks in the bush, so you may have seen a lot of these around. These are the fleshy brackets, so these are there are some really cool ones out there as well. And the ghost fungus, which some of you might have seen, it's often at the base of things like angophoras or even on deadwood as well, but they actually glow in the dark. And bioluminescence is an expensive trait to maintain, so it's not known yet why they have retained that trait, but it could be because they're attracting something like maybe a gum moth that spreads its spores. These are the earth stars and puffballs. So these things, they actually burst open at one of the points that you can see, like where there's a little stoma and a hole. And when the rain falls on to the ball part, little spores puff out of the holes, and that's how they disperse their spores. And it can look like smoke, like when there's a lot of them. It's a really interesting dispersal strategy. And these are the earth tongues. I'll just ask a favour now. If you guys ever see the green ones, please get in touch. I really like them, and they're very exciting. They're just so unlikely, and it's incredibly. It's just amazing to think that, you know, all these ones down the bottom that are all, they just look like black sticks, but they're actually all different species, and they all belong to us. They're from our biota. So these are all the ones that still remain in my yet to be named folder. I haven't been able to ID them. It doesn't mean that they don't have a name, but it means that, you know, I want to be able to ID them because I like categorizing things. And it just is an example of how much just in our area is yet to be discovered. So when you're out there and exploring, don't forget to look down. So I just want to introduce you to one more group of fungi. So these are the club and coral fungi. And this is the group that really caught the attention of Michael and myself. And it's because they, well, apart from being really beautiful, they actually span all of the different lifestyles. So they are saprotrophs, they're parasites, but they're also mycorrhizal. And we started to notice, you know, ones that looked similar were turning up in association with similar native plants. So we wanted to look into that more, but also look at the photos you can take of them. Like if you showed someone that, they wouldn't know if it was underwater or in the bush. And when you tell them it's in the bush, they go, "What, really?" So, and we like doing that to people. So, yeah, from here I'll hand over to Michael to tell you more about how we know the difference between these two purple fungi.
Thank you. Okay. All right, so I'm going to change gears a little and talk about some of the DNA work that we've been doing with these specimens. So here's the club and coral fungi. You can see why we might have chosen to concentrate on these. I mean, you can't look at everything, but we were just blown away by these. And it's also an advertisement for paying attention to the small things, because most of these things, they're no bigger than fist-sized, and most of them are only match-sized. So this is what we did. We crawled around in the bush on our hands and knees, getting lots of ticks and leeches, which isn't the fun part. We photograph them, we collect a specimen. This one here is Romeriopsis crocea, and that, for those of you who know these things, that's an achianthus leaf, a mosquito orchid, which are about the size of your thumbnail. So you get an idea of how big they are. We put them in ethanol for DNA, and we take all of the things that we meant to take, like GPS and environmental data and specimen descriptions. And of course, everything's collected under a scientific license. You can't just go out in the bush and collect stuff. Here's some specimens that we brought back for preservation, just to give you an idea of the kinds of things that you would see on any particular trip. All of these are then dried and preserved for submission to herbaria or fungaria, or what are they called now? Fungariums? Yes, fungaria. What we do is we extract the DNA and we use PCR, which you would have heard about with COVID. The reason you can diagnose COVID so quickly is this DNA technique called PCR. And what we do is we make billions of copies of a particular gene that tells us about the identity of these fungi. We then DNA sequence it, and then we do what's called aligning the sequence. And so you can see for these specimens here, all of which are in a genus called Romeriopsis, You can tell they're in Romeriopsis because of how similar the DNA sequences are here, and you can tell that there are different species because this group is different from that group, is different from that group, etc. So you can align these sequences, and that tells you both the identity of the thing that you're doing the DNA sequencing and its relationship with all of the other things that you're looking at. And that allows you to construct family trees. Here's a family tree of the Romeriopsis, and it's pretty complicated, so we're just going to concentrate on just this little bit over here, because these are two of our favourites, and they're the only two that we can reliably identify. It's Pulchella, which is Little Beautiful, here, and Crocea, and they're pretty distinctive. And so when you DNA sequence them, here's the family tree of the ones in the Lanco Valley. They're pretty close to ones elsewhere. Here's the family tree of Crocea from the Lanco Valley, and these are from overseas. So you can see all of our specimens are pretty similar, slightly different from the same species overseas. And that red line there is where you draw the species cutoff. So knowing these two species, we can say that's one species there, that's one species there. Here's another species here. What's this one? So this is an unknown species. From Brownsfield, it looks like, yeah. When you actually zoom back out to that tree that I showed you before, the circular one, that's this one, all of those other things there are all pretty much undescribed. So this is a summary. Well, I've just categorised them according to colour because it's easiest. So there's 4 brown ones, 2 to 5 unknown buff ones, 3 unknown white ones, and 7 unknown ones that look like this. And that's just one genus in the Lanco Valley. So that's Romeriopsis here. So in this plot, everything to the right-hand side of the vertical red lines is an identifiable species. Everything on the left-hand side is unidentifiable. And so there are 5 different genera of club and coral fungi, and you can see that maybe 80% of what we discovered is undescribed, unknown species. And that's just in the Lanco Valley. So this gives you an idea, harking back to what Ness was talking about the undiscovered and undescribed stuff, how much there is out there to still think about and just describe, not understand its biology or understand what it's doing in the landscape. So here's one of those genera, Romeria. These are the slightly larger clubs. They're usually fist-sized to maybe golf ball-sized, something like that. Most of these species, we've got no idea what they are, but we do know that they occur below or adjacent to particular kinds of trees. So this one, coachwoods. This one, peppermints. They're almost certainly mycorrhizal. In other words, they're forming an intimate association with the plants. And so therefore they're important. They're necessary for the trees to survive and for seedlings to thrive. So summarising the DNA data, we've looked at lots of different genera. The story is the same every time. Somewhere, maybe 50 to 80% of what we're looking at is undescribed. And these are 3 species that we've got no idea what they are. But what we can do is we can identify them down to genus. So you know there's genus and species. So we can identify the genera. And when we do that, we can plot how many genera we expect to find in a 10 metre square area and how many specimens we expect to find in that same area. And you can plot it out on a map of the Lanco Valley. And that's what we've done here. So each one of these dots represents a 10 metre square area, and what we've got are 4 hotspots. This is Coops Creek behind the Sanitarium Hospital. This is Browns Field, which is a Jurassic volcano. This is Sheldon Forest, which is high bluegum forest, and this is Blackbutt Creek and Falls Creek in Gordon. You'll notice that none of those hotspots, which are repositories of fungal diversity, are actually in the national park. And in fact, the national park has very poor fungal diversity overall. So that is a bit of a concern. What we've been doing, though, is just looking, as Ness said, at the fruiting bodies, at the corals and clubs that appear above ground, and they're the reproductive structures. But most fungi look like this most of their lives, and a lot of them never do this at all. So the only way of really getting at the extent of this problem is to actually examine fungi directly from soil. So the best way to look at fungal diversity is from soil. And why would we be interested? Well, while we've been walking around, we notice that the fungal populations, the kind of fungi you see, differ between what you might call pristine areas, not that there is such a thing, but the better bush areas have these in them. And they're all known to be mycorrhizal with trees. They support eucalypts. In weedy areas, and also importantly in bush regenerated areas, you see these fungi here. And those fungi are either saprotrophs, feeding decaying, dead material, or they are introduced, like the Amanita muscaria, your standard children's picture book mushroom. So what this suggests is that bush regeneration doesn't regenerate fungi that are essential for long-term health of trees, ecosystems, and bushland. So we can do our bush regen, but it's possible that those trees may never seed and produce seedlings in that area because the fungi are gone. So that's the question that we're really interested in. So, what's the fungal diversity in weedy, regenerated, and pristine bushland areas? And here's some examples: weedy, regenerated, and pristine. So what we did, we collected soil samples along 3 urban creeks in the Ku-ring-gai Council area: Cowan Creek, Blackbutt Creek, and Stony Creek in Gordon. And we designed the experiment like this. We run down the creeks and we take a sample of soil every 100 metres for about 2 kilometres, and we note whether it's pristine, weedy, regenerated, etc. This is Stony Creek in Gordon, where the flying fox colony is. And importantly, we know the history of the bush regeneration and weeding along these creeks. So, what we then do is extract all the DNA. We use PCR to amplify bits of fungal DNA that we can then sequence. And this is the really exciting part for me. I mean, when I first started in this business, if you got a DNA sequence, you could get a paper in Nature or Science, one of the biggest journals in the world. We now have this. This is called a nanopore DNA sequencer. And you'll see that in 5 hours, 7 minutes and 31 seconds, we got 2.72 million sequences. We're still obviously doing the analysis on these, but we've only done Cowan Creek so far, and there's at least 4,200 different species that we can detect just along Cowan Creek. So we're aiming to analyse about 200 of these soil samples, 54 sites, 8,000 or 8 million fungal sequences, and answer this question about fungal diversity. What we expect to have at the end is a survey of fungal diversity along these urban creeks. So how many species are there? How many are undescribed? How does soil properties correlate with fungal diversity? Because we're going to test the soil properties, nitrogen, phosphorus, pH, things like that. So does pollution affect fungal diversity? Identify differences in fungi from pristine, regenerated and weedy areas. And that's the main goal of this project. to eventually identify hotspots of fungal diversity that might act as sources of inoculum, where you can go out and dig up some soil, because we know there's lots of fungi there, and use it as part of bush regeneration to reintroduce these fungi that we might have lost during the degradation from native forest to weedy urban bushland. And it's going to come from places like this. Coachwood Gallery rainforest, much of which has been severely compromised by putting sewer lines down creeks. So the main places where we see really good fungal diversity are places without sewer lines. Okay. Oh, they don't all. They don't all. And sometimes the one side of the creek without the sewer line is better than the other side, yeah? Yeah, yeah. So just finally, just want to say, pay attention to the small things, because what COVID taught us when we were looking in the native bush, I mean, I've been in the bush all my life, but COVID really taught me to slow down and look at everything, because every time you go out, you find something new, every single time. And all of these are in a 5K radius of South Turramurra. There's some amazing things here, not just fungi, but plants. And so we've discovered rare plants and insects that haven't been described, and weird snails and strange parasites, and all sorts of things. And just before we go to questions, I'd just to acknowledge, mainly Ku-ring-gai Council Environmental Levy Grant and STEP from South Turramurra for funding this research, which is still going on, of course, and we've still got a lot of work to do yet. So thank you very much.